All right, hope everyone's doing well. Today we are going to start on Module 2 to review everything you're going to need for that module on the Keystone. So we're going to start off with cell reproduction today, and I'm only going to go over half of it. I'm going to go over cell reproduction that happens in our normal cells. So with this, I'm talking about your body cells. They're also called somatic, S-O-M-A-T-I-C cells. And these are going to be cells that help you grow, help you get into a more developed individual. Also, you go outside, you're playing, you fall, you scrape your knee, blood everywhere, okay? This type of cell reproduction is going to help to fill that hole that you just bruised with new cells. So that way it's helping in repairing your body. So as we are going to go through this, guys, I'm going to refer to the cell cycle a good bit. When we are going over the cell cycle, we're going to have two major parts. We're going to have interphase, and then we are going to have this area over here. It's called M phase, and that M stands for mitosis. And mitosis is simply a way that our normal body cells, like I just mentioned, reproduce, okay? So in other words, when you scrape your knee, we need to fill that hole with new cells. Right? We're not just going to space the cells out. We're going to make brand new cells to help to fill that hole. All right. So let me start off with interphase. Interphase is going to be the time when the cell is not dividing. Okay. But it could be getting ready to divide. So this whole area right here, in most cells, and I know this looks like 75% based on how I did this pie chart, it's actually about 90% of a cell's life. And it's going to be divided into three phases. So we're going to start up here. This is going to be called G1. And what's going to happen here is our cell that is a brand new cell, it just got done being made, our cell is going to grow. And it's going to get bigger and bigger and bigger and finally reach maturity. Now, after the cell gets so big, it is going to be a completely developed cell. So it's kind of like you guys, once you develop enough, you're able to reproduce. Your cells are actually the same way. So once it grows, once it reaches maturity, now it can focus on reproducing. And this is going to start our reproduction process here. So this one is going to be called S phase. S stands for synthesis. All we're doing here is we are copying our DNA. We're copying our DNA. Why are we copying our DNA? Well, let's just talk about human DNA to begin with. Each one of your cells, your normal body cells, has 46 chromosomes. We want every new cell produced to also have 46 chromosomes. So what we're going to do here is we are going to copy that DNA. So now, instead of having 46, we are going to temporarily, not all the time, temporarily have 92 chromosomes because we made a copy of it. We duplicated it. We went from 46 times by 2, you get 92. All right. Then our last phase over here, down here, this is going to be called G2. And the G stands for gap, okay? What the cell is going to do here is it's going to continue to grow, but also we are going to check that DNA. Why are we checking the DNA? Well, we're checking the DNA to make sure it's free of mutations. We do not want this brand new cell that we are making to have tons of mutations in it and possibly cause the cell to not do its function, or even worse, cause some type of cancer. Now your cells are really, really, really good in making sure there's no mutations, and if there is, checking them, making sure that there's not, if there is, labeling that and actually fixing that mutation, okay? So interphase. 3 parts. G1, we grow. S phase, we copy our DNA. And G2, we check the DNA. Make sure it's free of mutations. Now we're going to go into our M phase. This is going to be where the cell is actually going to reproduce, where it's going to make a brand new cell. So we're starting off with one cell here. By the end of M phase, right in here, we should have 2 new cells. Yeah, they're going to be little because that's why we got to have G1 again for it to grow, but we will actually have 2 distinct cells, okay? So I'm going to start here. Our first step of the actual cell reproduction process is called prophase. Now, before I go over prophase with you, we have 46 chromosomes, as I talked about, and when we duplicate them, we have 92. Where do you get them chromosomes from? Well, you get one from your mom, you get one from your dad, okay? So here's what I'm going to do here. We're going to say this is chromosome number 1. And this will be chromosome number 2 here, okay? So let me just start off with number 1. And we're going to say this chromosome in blue is from your dad. This chromosome in red is from your mom, okay? Now let me move over to chromosome number 2, and I'm going to use some different colors so we don't get too confused here. We're going to say the brown one is from your dad. The purple one is from your mom, okay? So the first one is from your dad. I just decided that order. It's no particular order there. And the second one is from your mom. Now, this right here shows I got 1, 2, 3, 4 chromosomes pictured. I have to get all the way to 46. I'm not writing them all right? We're not going to go all the way to drawing 46 chromosomes. Here's what I want to mention with this, though. This is chromosome 1, you have 2 of them. This is chromosome 2, you have 2 of them. If I drew chromosome 3, you'd have 2 of those. We would have to go all the way to number 23. So in reality, you have 23 pairs of chromosomes. What these are called here, okay? Number 2, we got 2 of them there. One from mom, one from dad. Those are called homologous chromosomes. It's chromosomes of the same number. They are different because one's from mom and one's from dad, okay? So I'm only going to use those 2, but I want you to know that there will be, 23 pairs. So here's what's going to happen in prophase. So this is our first step. What we're going to do in prophase is I'm going to draw the nucleus, because this is a eukaryotic cell, and I'm going to draw it fairly large so you can see it. Here's our nucleus. I'm drawing it as a dotted line because the nucleus actually disappears here. Now, I'm also going to draw chromosome 1 and 2 in, but I'm going to draw them a little bit different than you see them up there. So I'm going to draw them in pairs together. So I have 2 brown ones, 2 brown chromosomes. I have 2 purple chromosomes. I also am going to have 2 blue chromosomes here. And I'm going to lastly have 2 red chromosomes. Well, you're probably like, why you got 2 now? All right, we only got 1 of each over here. Why do I have 2 over here? Here's why. Remember, in S phase, we copy our DNA. So you're copying dad's chromosome for number 1, you're copying mom's chromosome for number 1. So now you have 2 of them, and that's why you guys see chromosomes always pictured as little X's, because we copy them, all right? So anytime you see chromosomes as X's, it's after S phase has already happened, right? So, prophase, nucleus disappears, that's your first thing. Second thing, the chromosomes, your DNA, it condenses to turn into this chromosome structure. Before this, it looks like spaghetti in a bowl. It's called chromatin, T-I-N at the end. And when that chromatin condenses, it becomes chromosomes. And again, that's what happens during prophase. The other thing we are going to have, and I told you in a previous video, we will talk about these later on, these are called centrioles. And I'm going to show you what they are going to do, but they're going to slowly move toward the poles, the different parts of our cell, the different, like North Pole and South Pole. I'm making them go east and west. All right. That's it. That's all that happens during prophase. Nucleus disappears, chromatin condenses into chromosomes, centrioles move outward. All right. Let's go to metaphase. This is going to be our second one here. So let's start off with our centrioles. They are now completely toward the poles. Here's how to remember this one. Metaphase means middle. These chromosomes are going to line up in the middle of the cell. So they're going to form a nice line. Now they could go north and south, going up and down. If your centrioles are north and south, then these chromosomes would line up across, I guess we can call it the equator there. All right, let's draw the purple one. And then lastly, the brown one. Cool. The last thing we are going to do in this part is we are going to connect something called spindle fibers. And these are microtubules that are going to come out. It's like Spider-Man coming out of the centrioles here. And they are going to connect to our chromosomes here. So these are called, like I said, spindle fibers. There we go. All right, we're connected. Now, anaphase, this is going to be number 3, our third part of mitosis here. The spindle fibers and the centrioles, they work together. So it's almost going to be like a fishing rod. You know, you cast out, you're going to maybe catch a fish. When you catch that fish, you're reeling it in. So here's what's going to happen. The centrioles are going to start to pull in the spindle fibers. Now, if we look over here, it's connected to both sides of our chromosomes. So here's what happens. It literally yanks the chromosomes apart. So I'm drawing them as halves. In reality, what's going on is they are returning to this structure over here. Okay? Brown's next. And lastly, purple. Okay. So that is anaphase. So metaphase, they lined up in the middle, spindle fibers attached. Anaphase, the spindle fibers got pulled in closer to the centrioles, the chromosomes separated. So now we have one chromosome on each side. What we call these are chromatids, T-I-D at the end. Don't confuse that with chromatin, T-I-N. I know it's literally one letter, all right? Whoever made that part of the English language just tried to screw with us there, because they literally made 2 words off by 1 letter, and they mean something so similar, but so different. So chromatin was the uncondensed DNA. Chromatids are after our chromosomes become separated, all right? So I'm going to draw telophase a little bit different here. So what happens during telophase? Is our nucleus is going to reform. Now our cells aren't completely separated yet, but they're close. You guys can see that there's a little dent in the middle there. So this cell, this is going to be on this side, is going to have one of our blues. This cell, one of our blues as well. We're going to have a red on this side, a red on this side in that nucleus. We're going to have a brown on each side. And then lastly, let's bring our purple in. Okay? And what will happen, guys, after this, we are going to separate the 2 cells into their own individual cells. Okay? So once they are here, like I said, they're not completely separated yet. Our 5th step, which I'm not going to really go over too much, I'll draw it over here. Right after telophase, we are going to have something called cytokinesis. It's before interphase and right toward the end of mitosis. What's going to happen here? Those cells separate. They become 2 distinct cells, okay? Also during cytokinesis, we split the cytoplasm up in between the 2 cells. We divide it equally, so they are very small. You don't have just one big cell and one little cell. They're both the same size, and then they will, once they hit G1, grow again, okay? All right, so that is it for mitosis, the whole process of it. The last thing I want to go over before we call it a day with our mitosis video is what types of cells does mitosis produce and some characteristics about those cells. Okay, so first thing, these cells, these 2 cells produced are 100% identical to one another. They're identical cells. You guys can see I got a blue in each, I got a red in each, I got a brown in each, and I got a purple in each. So they are identical cells. The second thing is they are diploid cells. What this means is I have 2 of each of our chromosomes. So diploid, if you're looking at number 1 here, I got a copy from mom, I got a copy from dad. The opposite of that, which we'll talk about next video, is called haploid. What would happen in haploid is we would only have 1 of them instead of 2. So a little bit different there, okay? Third thing, it produces body cells. Oops. And as we said, these are called somatic cells. And lastly, how many cells do we form at the end? 2. Okay. So that is the characteristics of these cells produced by mitosis. We want them to be identical because our goal is to have these cells, like I said, if you skin your leg, we want those cells identical to the cells that we lost. So this is the process of mitosis, guys. Next time we will go over meiosis, which is a little bit of a different process. We will go over how that one relates to our sex cells and reproduction. Have a good rest of your day, guys.